willkommen zur Dresdner Langen-Nacht der Wissenschaften, hier am Max-Planck-Institut für molekulare Zellbiologie und Genetik. In diesem Jahr virtuell. In diesem Clip wird euch Dr. Grapin-Botton, eine unserer Direktorinnen, über ihre Forschung mit Mini-Organen, sogenannte Organoide, erzählen. Dann begleiten wir sie noch in ihrer Labor und werden mit einer ihrer Doktorandinnen sprechen. Viel Spaß!
Hello, my name is Anne Grapin-Botton. I'm originally from France. I've worked at the Max Planck Institute for about 3 years. And what I brought with me was the technology of organoids. So organoids, you may have heard of it. You may have heard of humanoids. Humanoids are a little like humans, but not quite. So organoids are like organs, but not quite. That's the best we can do by growing cells in vitro in a dish to try to build organs. So to build these organs, we use what we know of normal development, and we try to make the cells do what we want. Organoids have been known for about 10 years. They're very useful in human medicine to do disease modeling. They're very useful also to do drug testing, to find cocktails of drugs that are targeted and very efficient on the patient and his disease. We use them to understand development because we can see the organoids. They are in a dish. So we work on pancreas-Organoides. We start from stem cells, we engineer these stem cells, and then you see the cells and they start growing. They self-organize. So self-organize is a bit mysterious, but you've seen self-organization. It's actually beautiful. You've seen the bird flocks, where each bird is part of a ballet, and they do these flight changes. The birds have very specific rules they follow, looking at their neighbors, but together they make an ensemble, and that's what the cells do. So what we did recently, we've worked for a long time on mouse organoids, but recently we've engineered from scratch, from stem cells, human pancreas organoids. And the difficulty when you do that is finding a recipe first, finding a recipe other people can use, but it's also making sure that what you have is real. And to test that, we actually compare the cells to donated human embryos. And then what we've done, we've been able to use this system in a dish to understand how human development is done, which is a process that's normally hidden. Okay, now let's go together to the real lab.
Rashmiparvathi Keshara. I'm a PhD student in Grapin-Botton's lab. And this is my second year as a PhD student in Dresden. So I did my master's and all my education in India. So I'm basically from India. And I came to Dresden right after my master's. And it was my first time outside my country. And I would say people in Dresden are really welcoming. And it's a very beautiful city. It's a very nice place, and I'm working as a PhD student in MPI-CBG here, and it's really an amazing place to do research. So as a PhD student, what I found interesting was it's a very nice city for education, for research, and also for social activities. So, as a biologist, I'm interested to understand how single cell divides and forms an entire organism. So in Anne's lab, we are working on pancreas development, and we use different models like organoids to understand development. So what I do is I differentiate human embryonic stem cells into pancreatic cells, and we make organoids out of it. So this is like organ in a dish. So we have different cells of the pancreas that we try to grow in a dish. So what we do is we have pancreatic progenitor cells that we form spheroids, and we try to differentiate them into different pancreatic cells like insulin producing beta cells or glucagon producing alpha cells and so on. So in my project, what I want to do is I want to treat these spheroids with different biomolecules or some compounds and see how they differentiate or what happens to these type of cells. So I will do a screening with different biomolecules and see what regulates the differentiation and development of pancreas. So what I do is I collaborate with different people in CBG, and we also collaborate with mathematicians and physicists and try to understand development of human pancreas.